Hello, we are doing eukaryotic cell structure now. Okay? If you've read the textbook, there's 4.4 through all the way through 4.22. I mean, there's a whole lot of chapter content on this particular topic. We're not going to attempt to cover it all in that amount of detail. I mean, it's kind of silly from my perspective that prokaryotic cell structure in chapter 4 has only 4.3 and eukaryotic cell structure has 4.4 all the way to 22, right? And so while we needed to supplement a little bit of the readings for the prokaryotes, we don't need to do that. In fact, I need to streamline things a bit for eukaryotic cell structure because truth be told, not every biologist is going to be so enamored with the details of cell structure and what the differences are between the smooth ER and the rough ER. Those are important points for some people. I like to take a much broader perspective view. In other words, I want you to be able to answer the big questions now, because later on, when you take physiology or anatomy, you're going to be getting into a lot more of those detailed issues with regard to the cellular organelles, right? We'll be doing some of it, but I think I want to start with these bigger questions. For example, right, in the previous chapter, we said that prokaryotes, bacterial cells, prokaryotes are small, and therefore they have an advantage of surface area to volume ratio, right? The general idea is that if you have a small cell is going to be able to double in size, right? You're going to be able to produce two small cells of the same size. They can basically double their biomass much faster than a larger cell could, because a larger cell is going to have to feed all of this volume. You've got a large volume relative to the amount of surface area of the plasma membrane that's there, right? And so that advantage of prokaryotes might make it seem like there's no reason for a cell to be larger. Eukaryotes would have to be at some disadvantage, right? And so I guess the question is, well, it must not just be a bad thing to be larger. There must be some advantage of being larger as well. And that's absolutely true. And you don't have to think too hard in order to get this. And you're not going to find this in the textbook, but it's worthwhile for me to share. Now, if you're a small cell, yeah, you can increase much quicker, but that's presuming, right? This here presumes that we have resources. We're presuming that there's plenty of food out here so that the bacterium can absorb the food much more quickly than the larger cell, and thereby convert those nutrients into biomass, and thereby increasing the overall biomass of the culture, right? Okay. Now, change that a bit. What would happen if you didn't have those resources, right? If in the absence of resources, the smaller cell would be kind of limited. I mean, it's not going to be able to double as rapidly. In fact, it might not be able to double at all. I mean, if there are no resources around for the cell to consume, then the cell is obviously not going to be growing. Okay, but it's also going to be at a deficit. I mean, if you say no resources, okay, if you were to put those cells into clear water, how long would they last? I mean, they would have to persist on whatever resources that are there inside of the cell, right? And once those resources are consumed, and there's no more energy left inside the cell for it to do what it needs to do, it dies, right? Now, the larger cell has an advantage of monopolizing a greater amount of resources. And so if you were to put a culture of larger cells into a medium without any nutrients, like with water or saline, right? If you just starvation conditions, the larger cell will persist better. because it monopolizes more of the resources, right? And so, yeah, it's not simply surface area to volume ratio is certainly an advantage of the smaller cells, right? Smaller cells, when under those conditions, when resources are abundant, the smaller cell could increase its biomass, it can increase its population by much faster, but with a much faster rate compared to the larger cells. However, under conditions of resource scarcity, a larger cell would have the advantage of being able to withstand those periods, right? So yeah, I think we've answered that question now. There is a advantage of being larger, okay? And the other thing about eukaryotic cell structure, it has to deal with the fact that in a larger cell, you really have lots of space. And whereas if you're in a small cell, you don't really have a whole lot of space to work in. So everything has to happen within that small compartment, right? If you're a larger cell, you could potentially split up the work into different zones of the cell. You could have one compartment doing energy processing. You could have one compartment doing manufacturing of proteins. You could have another compartment doing the breakdown of wastes, right? You have another compartment doing the control of information, right? And so the idea that we develop with eukaryotic cell structure, in fact, the theme of eukaryotic cell structure discussions is going to be compartmentalization. When we think about the organelles, they're basically cellular compartments, little membrane, usually membrane-bound areas, zones within the cytoplasm where specific functions will be taking place. And because we're putting these specific functions into localized areas, we can have those functions take place more efficiently than they would if they had to be scattered throughout the cells, right? And so this is kind of like our overarching theme of eukaryotic cell structure. And now we can get to the textbook discussion of the actual eukaryotic organelles. Ready? Okay, you don't have a choice. I'm going to do this anyways. Okay, so here is a picture from your textbook showing your prototypical eukaryotic cell, nucleus, nuclear envelope, nucleolus. You got ribosomes out here, then rough ER, smooth ER, Golgi, mitochondria. So it's one thing to look at this and say, okay, yeah, these are the structures. Okay? It's another. You might be already be thinking this way, but to me at least, I need to think one more level deeper and realize that the reason why we have a nucleus is that all the nucleus stuff, all nucleus-like function okay. I could say the nucleus functionality is localized within the same area, and therefore we could have a more efficient achievement of the objectives of whatever the nuclear function is, right? Whatever the function is that's carried out by the endoplasmic reticulum, this corresponds to a space within which all those functions can be localized with a much higher concentration of the reactants. And so we can get a much higher conversion rate of the reactants into products, right? And so all of these spaces, whether we're talking about a space like this or a space like the mitochondrion, which we have energy processing taking place, we can see that the structure of a eukaryotic cell is based on the theme of localizing those specific functions within particular areas. Okay. Now, there's more that we can be discussing within this particular section. There's some generalized, there's some videos that you can look through. They're not terribly interesting. But what I want to do right now is to skip all the way to the last portion that I'm having you read, right? Which is this review section, okay? And at this point, you should have already read through the chapters, right? And so what you get at the very end is a really cool and neat and convenient way of organizing your understanding of the structures within eukaryotic cells. You have four basic functionalities, four basic categories of functioning. And you could place the cellular organelles within one. There might be some overlap, but they could mostly be placed within one of these particular areas of cellular absolute necessity, right? For example, a cell needs to manage the information that's there in the cell. We need to keep that genome, that DNA genome, organized. We need to make sure that it's replicated appropriately. We need to make sure that it's used appropriately. We need to be able to get those genes converted into RNA so that they can go out into the cytoplasm and become protein. And so all of that stuff is kind of a related functionality, and most of it is localized within the nucleus, right? And then you look at the endoplasmic reticula and the Golgi apparatus and the lysosomes and the vacuoles. And we actually say that they're part of the same general organelle system. We call it the endomembrane system. But when you look at the functionality, they're all related to the synthesis and manufacturing and processing and distribution of gene products. Okay? So the proteins that are being produced by a cell for export out of the cell. Okay? Secretions. When you have a protein that's going to be making lots and lots of either secreted enzymes or secreted antibodies, these are cells that are going to rely heavily on their endomembrane system organelles, rough ER, smooth ER, and Golgi, right? And so if you think about the functionality of those parts, all of those organelles kind of fit together into the same little cubbyhole. In other words, it doesn't really make sense for you to think of the rough ER and mitochondrion as being related, even though they're subcellular organelles inside of both animal and plant cells, right? No, mitochondria has its own functionality. It's related to energy processing. And in plants, we also have chloroplasts, right? And we get to this later on in this unit. Very soon, we'll be talking about ATP synthesis and how cells are generating the ATP energy that they're using at all times. And so the mitochondria is going to come into that. But mitochondria, even though it's a membrane-bound structure, is actually, it might look superficially kind of similar in some ways to the Golgi, okay, but it's actually coming from a different source, and the origin of mitochondria and chloroplasts is what we'll be developing in the next lecture, right? And finally, there's the cytoskeleton, okay? I call it a cytoskeleton in my outline, but the book identifies it as structural support, movement, and communication between cells. But we're putting a lot of things in there, and we're not going to be making a big deal out of this, although the concept of a skeleton, right, is something that you normally think of as something that supports your body and it also allows you to move around, right? So skeleton, your skeleton is what kind of defines your shape, right? If you didn't have bones, for one, you wouldn't be able to keep much of a shape, right? You'd be more like Jabba the Hutt, okay? Moreover, if you didn't have a skeleton, you really wouldn't have any efficient way of moving around. Okay, you move through your world by moving your bones. Okay, and wherever your bones go, your skeleton goes, you have to go along with it. In other words, cytoskeleton has those same analogous functionalities because, at least for animal cells, it's going to support a cell, making it look the way it does. We'll be talking about different kinds of cells later on, like nerve cells, those nerve cells that have long extensions, they call them axons. So why is it that some cells look like this? Long axon. So this might be a nerve cell, whereas a liver cell might just be looking like this. And a skin cell might be looking like this. In some cases, the shape of these cells, the specific shape of these cells, is held in place by a framework of elements, the cytoskeleton, right? And that same framework is related to the structures that will be moving cells when you actually have cells that could swim. For example, paramecium, there's a picture of that in your textbook, is covered by little cilia. And these cilia are beating the water across the cell surface. And the cell is able to swim around. The components of the cilia are the same components that we find inside of the cytoskeleton. And so it's kind of, it is, to me, kind of neat to think that, we've got something occurring at a single cell level that is more or less parallel and analogous to what we have at a whole organismal level in the human body. Okay. Now, let's see if I can go through this without having to go back to my original outline. And so I'm going to scroll back up and just kind of tour through this chapter with you, going through some of the things that we'll be focusing on. There's more detail in the textbook than what I'll be holding you responsible for. Okay, but it's still it's definitely worth your while to go through and read these sections of the textbook very carefully. Right. So there's a section on nucleus telling you what happens in a nucleus. Right. So you've got the storage of DNA. You've got the synthesis of RNA. Okay. And there's a specific structure, the nucleolus, right? It's darker structure within the nucleus, right? Nucleolus means little nucleus, right? And so it's like a nucleus of the nucleus. But we know more than just that it's a little spot inside the nucleus, right? At one point, that's all it was, a little spot that we could see inside the little spot that you found inside of cells, okay? But now we know that it's the place in which we have the synthesis of the RNA and proteins that are going to be making up the ribosomes, right? So ribosome synthesis is going to be associated with a nucleolus, and that's going to be something that we'll be making reference to later on. Okay? In 4.6, it talks about ribosomes, and ribosomes are going to be of great discussion in our last unit. Okay, so we're past exam 1, exam 2. For the material for exam 3, we'll be talking about the central dogma and how ribosomes are going to be doing exactly what you're seeing in this picture, converting a messenger RNA molecule's information into a sequence of amino acids, which will create the polypeptide, will be making up a protein, right? And so this is your opportunity to think carefully about ribosomes before it's kind of like planting a seed in your mind so you can refer back to this anchor point later on in the semester when we actually talk about these things, right? The next few sub-chapters are going to be related to this thing I mentioned it before, the endomembrane system, right? Endo, inside, membrane. These are all membranous organelles, including the two parts of the endoplasmic reticulum. And there's a better picture of it in the next section, where you've got the rough ER. Those are the ones that are associated with ribosomes. Ribosomes make it appear kind of granular, which is why it was called the rough ER. Then you've got a smooth ER where you don't have the ribosomes. But remember, ribosomes are making the proteins. The proteins are going into the space inside of the rough ER, and those proteins eventually make their way out to the smooth ER, where they're processed further, eventually getting sent over to the Golgi, where they're processed and packaged even further. It's a little protein factory, this endomembrane system, right? Golgi is doing further processing and packaging. Then you have things like lysosomes, which are a good example of the actual particles that, the little vesicles that are being produced by the Golgi. Right? Now, in the next lecture, you're not going to read this for this particular video, but in the next lecture, we'll be talking about the mitochondria and the chloroplasts. Well, in the next lecture, we'll be talking about the origin of the mitochondria and the chloroplasts. And in coming videos, we'll be detailing what's going on in them. But basically, the idea is that both mitochondria and chloroplasts are the places where we have the synthesis of ATP energy, right? What's going on in the mitochondrion? What goes on in the mitochondrion? ATP production. What goes on in the chloroplast? Well, ATP production. Oops. Next slide. What goes on in the chloroplast? Two things, actually. ATP production. Okay. And don't get me wrong. Plant cells also have mitochondria. All eukaryotic cells have mitochondria. It's that chloroplasts, plants also have chloroplasts in addition to the mitochondria, and chloroplasts also contribute to ATP production for the cell, right? But in addition to ATP production, we also have photosynthesis. Which is the production of sugars from CO2 and water. In other words, we're using light energy to convert CO2 and water into sugar. You want C6H12O6, and we're also getting O2, oxygen gas. Right? In other words, chloroplast and mitochondria are definitely related in the sense of being energy processing plants for the cell. And consequently, they're also, we classify them together. Now, the last portion of the chapter is going to be focusing on the cytoskeleton elements, the parts of the cells that are going to be responsible for giving the cells its shape, as well as its ability to move around. I'm not going to include that in this lecture. The one thing that I will point out at the very as kind of like a side note that really can't be ignored is the fact that plant cells, not animal cells, plant cells have it's not even a good picture. They have cell walls that are outside of the plasma membrane. So let me do this a little bit more clearly. It's not exactly the picture that I wanted to show you. So right here, oh darn it, draw picture. Do this blue. Just look for the blue line here, right here. Etc. That's the cell membrane, plasma membrane. We've talked about that. That's the phospholipid bilayer together with the proteins that transport stuff across the plasma, the cell layer. Okay? Now, I guess the point here is that in plant cells, we have to have little connecting tubes to connect this cytoplasm with the cytoplasm in the next cell, right? So there are little tubes. These are the plasmodesmata that connect cells one to another. The thing that I want you to think about here, right, is this brown layer outside of the cell membrane. And it's going to be made up of layer after layer of cross-hatched cellulose, and that's going to be the cell wall. Cell wall is outside cell membrane. Okay. It's actually a big mistake that students make a lot, all the time, which is to think that the cell wall is somehow the same as the cell membrane. Okay. No, it's not. Okay. Don't confuse the two. If you mean cell membrane and you write cell wall in your exam, that's not going to give you any points because the cell wall actually is a structure and it's different from the cell membrane. It's something that happens outside. Cell wall is rigid. Okay. Cell membrane is our phospholipid bilayer. Remember phospholipid bilayer? Remember what phospholipids are? They're just like little molecules that have a phosphate head, a hydrophilic head, and two hydrophobic tails. Okay, and that's principally what the cell membrane is. There's nothing holding it tied up, fixed in any way to the next phospholipid that's sitting next to you. These two phospholipids can move around relative to each other. In other words, it's a fluid structure. Cell membranes are inherently fluid in structure, whereas cell walls are rigid, usually cellulose material. We talked about that when we discussed polysaccharides, right? So that was like a little warning shot across the bow. Make sure that you be a good biologist and don't make the mistake of confounding the phospholipid bilayer, the cell membrane with the cell wall. Okay, so let's kind of go through this, check off everything that I wanted to talk about. Compartmentalization gives us greater efficiency because we're specializing functions within localized cellular spaces. We've got these four functionalities. I'm not going to focus on the fourth one because we're trying to streamline this content, but we do have areas that we will return to later. The information management, when we get a chance to talk about molecular genetics, we'll be coming back to this, and we'll be making reference to the organelles that are part of the cells responsible for that area of functionality in cells. Okay. Manufacturing, distribution, and breakdown is the endoplasmic reticula, the Golgi, the vesicles, vacuoles. Okay. And since we do address things like the effector B cells, the cells that produce lots of antibodies, that's a process of manufacturing and secreting proteins. And so we have contextual examples later on in which I'd like to be able to have you recall what we talked about in terms of the organelles of cells that are responsible for that protein secreting functionality. Okay. And very soon we'll be talking about mitochondria and chloroplasts and how they contribute to meeting the ATP needs of cells. Okay. So we're not going to focus on this. Don't worry so much about cytoskeleton, except for the fact that we have cell walls in plants and fungi. It's extracellular, rigid material cages around what's otherwise a fluid cell membrane. But in animals, we don't have a cell wall. The shape of an animal cell is coming from the internal cytoskeleton. Okay, that's it. We'll be talking to you about the origin of mitochondria and chloroplasts next.